Hello students, in this video we'll be covering gluconeogenesis, that is the process of synthesizing new glucose. Now, why glucose synthesis is required? So, glucose is a major or sole source of energy for tissues like brain, nervous system, testis, renal medulla and embryonic tissues. In animals, the gluconeogenesis process takes place mainly in liver, and some of it also takes place in renal cortex, that is in kidneys. Now, animals make glucose from compounds like lactate, pyruvate, and glycerol. Plant seeds make sucrose from stored fats and proteins, and microorganisms they can make glucose from a variety of compounds like acetate, lactate, and propionate. Now, a bit of comparison between glycolysis and gluconeogenesis. So they share 7 out of 10 reactions. Now, in case of glycolysis, the reactions catalyzed by the three enzymes, that is hexokinase, phosphofructokinase 1, and pyruvate kinase, are irreversible. And to bypass these steps, three separate enzymes are used in gluconeogenesis. Now, both pathways take place mainly in cytosol, and they are regulated separately through unique enzymes of each pathway. So now we'll look at the three reactions of gluconeogenesis that are different from the glycolysis. The first one is pyruvate to phosphoenol pyruvate. So we are the last step of glycolysis is going in reverse, but through different enzymes. So first, the pyruvate is transported to mitochondria, where it is converted to. This is the overall reaction where pyruvate plus ATP plus GTP plus bicarbonate gives phosphoenol pyruvate plus ADP plus GDP plus inorganic phosphate and carbon dioxide. Now, first, pyruvate is transported to mitochondria where it is converted to oxaloacetate by the enzyme pyruvate decarboxylase. Now, this enzyme is allosterically activated by high amounts of acetyl CoA. Now, what can lead to high amounts of acetyl CoA is if the breakdown of fatty acids is already going on, then it produces high levels of acetyl CoA, which means that another energy source is available and glucose breakdown is not required, and the pyruvate can be used to production of glucose. Now, this oxaloacetate is not directly transported out of mitochondria because there are no transporters for oxaloacetate. So it is first converted to malate by using NADH to reduce oxaloacetate, and this malate is then transported from mitochondria to oxaloacetate. Malate is transported from mitochondria to cytosol, where it is again converted back to oxaloacetate, thus producing NADH in the cytosol. Now, in the cytosol, the oxaloacetate is converted to phosphoenol pyruvate by the enzyme PEP carboxykinase. It is phosphoenol pyruvate carboxykinase. And this enzyme uses GTP instead of ATP for phosphorylation. Now, when lactate is the starting material for glucose, what happens is lactate is first converted to pyruvate in the cytosol of the liver cells, and this produces NADH in the cytosol. And this pyruvate is then transported into mitochondria where it is converted to oxaloacetate, and this oxaloacetate is directly converted to phosphoenol pyruvate by a different enzyme, that is PEP carboxykinase, but located in the mitochondria. So the carboxykinase enzyme has two versions. One is located in mitochondria and one is located in the cytosol. Now coming to the second unique reaction, that is conversion of fructose 1,6 bisphosphate to fructose 6 phosphate. Now, as we saw in the video about glycolysis, that this is the committing step of glucose to glycolysis, that is conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. And so it has a high free energy change. So to bypass it, a different enzyme is used in this case. So fructose 1,6 bisphosphate is converted to fructose 6 phosphate by the enzyme FBPS1. Now, when fructose 6 phosphate is converted to bisphosphate, ATP is converted to ADP, but in the case of this reverse reaction, there is no production of ATP. The phosphate which is removed is just released in the form of inorganic phosphate. Now, moving to the third reaction, that is conversion of glucose 6 phosphate to glucose. Now, this reaction is carried out by enzyme glucose 6 phosphatase, and again, similar to the last reaction, there is no formation of ATP, but the phosphate is released in the form of inorganic phosphate. Now, this final enzyme, that is glucose 6 phosphatase, it is absent in brain and muscle cells, and that is the reason that these cells cannot perform gluconeogenesis on their own, and they have to depend on the glucose produced by liver or kidney as an energy source. Now, energy balance of gluconeogenesis and glycolysis. So this is the overview of gluconeogenesis, and in the whole process of converting the pyruvate to glucose, the total free energy change is minus 16 kilojoule per mole, and this is the equation: 2 pyruvates plus 4 ATP plus 2 GTP plus 2 NADH plus 2H plus 4 H2O gives 1 glucose plus 4 ADP plus 2 GDP plus 6 inorganic phosphate and 2 NAD plus. And in case of glycolysis, the total free energy change is minus 63 kilojoule per mole, and the overall equation is glucose plus 2 ADP plus 2 P inorganic phosphate plus 2 NAD plus gives 2 pyruvate plus 2 ATP plus 2 NADH plus 2H plus and plus 2 H2O. Now, by comparing both of these reactions, we can see that when glucose is converted to pyruvate, we only get 2 ATP molecules and 2 NADH per glucose. But when the reverse is taking place, that is, the pyruvate is converted to glucose, uh, total 4 ATP and 2 GTP are used, and 2 NADH is also used, and there is no production of ATP. So, energetically, this is a very expensive process. But since the brain and nervous system mainly relies on glucose as energy source, this is required. In the next video, we'll see how both of these pathways are exactly regulated, because as we learned in this video that 7 out of 10 steps are common in both pathways. So if these pathways are not regulated properly, it will just lead to a wastage of ATP and NADH. That is the wastage of energy. So how these two pathways are exactly regulated, that we'll see in the next video. Thank you.